I recently attended a departmental research seminar, and the speaker described a study that sought to identify microbiome effects of a treatment in mice. There were 20 mice in a total of 4 cages. Each cage was randomized to receive one of 2 treatments, either antibiotics delivered in the water or just plain water. At the end of the treatment period, they did microbiome analyses. As she showed the results, she noted that the control mice were very tight, with little variability. However, in the active treatment, the mice seemed to cluster into 2 different groups. The audience of other faculty and residents began hypothesizing about why such a difference could occur, but I asked whether the results clustered by cage. In fact, they did. The speaker had group housed the mice, so 5 animals in each of 4 cages. What was different about the cages is not entirely clear. Gut microbes have been shown to be altered by a large array of factors, and certainly a shared environment would encourage sharing the microbiota. For instance, one group of mice may have consumed more antibiotic water than the other group of mice, but consumption was not quantified in the study. In addition, in one of the cages, a mouse was diagnosed with a dry tail, and an appropriate animal care standard for treating it was application of an improved moisturizer. Mice are coprophagic, meaning they eat feces. If there was something different about a mouse or mice in one cage, it could spread to others within the cage. I could think of no better example of the challenges of group housing animals than this, particularly when the outcome can so easily be contaminated between animals. In the end, she essentially had n equals 2 in each group, 2 cages. Mice were not independent, only the cages were.
So how can we learn from these mistakes? From a statistical point of view, when treatments are applied to cages instead of individual animals, individually housing animals would provide the greatest power. It maximizes the number of experimental units. But, of course, how animals are housed depends on more than just statistics, including animal care protocols, the design of the experiment, and the exact question being asked. There are at least 3 things to consider, then, when conducting experiments with group-treated animals. First, recognize that the unit of randomization is the group, not the individual mouse. Second, when designing the experiment, recognize that power and sample size calculations are functions of cages, not just mice. The number of cages typically has a much larger effect on the power of the study than does the number of mice. Third, be sure to incorporate cages and group housing into the statistical model. Otherwise, the statistical analyses may tend to reject more null hypotheses than they should. And of course, if you have any questions, consult your friendly neighborhood statistician.